So, all right, very good. So let's go ahead and get started. It's good to see everyone now that I've figured out how to do that. Thank you all for joining. I've really been looking forward to doing this speaker series Zoom meeting. Our speaker is Dr. Paul Nelson, and I'll do a more formal introduction of Paul in just a second, but I just a couple of comments to precede his talk. And I know some of you have probably heard me say these things before, but I truly believe that we really do live in a privileged time. And I say that from especially the past 50 years in the context of the scientific and technological advances that we have seen, just in particularly if you're as old as I am, that we see advances that are pointing us toward God instead of away from God. And that may have always been the case. In fact, I'm sure that it was always the case, but it seems that it's even more so now, perhaps, than it ever has been. I mean, you think about our understanding of the universe and the vastness of the universe, and we can now look back to within like 500 million years of the Big Bang with the technology of the Hubble Space Telescope, advances in microscopy like electron microscopy. We can see the complexity and the intricacies of the microscopic world. We've sequenced the human genome, and we have got a better understanding of information required for all biological systems, and we understand that even Darwin's simple single cell required a vast amount of information for it to come into being. So I mean, it's got to be an incredibly exciting time to be working in the field of intelligent design, and our speaker's been doing that for a career. I'm going to read Dr. Paul Nelson's bio because it's so good, and I don't want to mess it up, and then we'll get into his talk. Paul A. Nelson is a philosopher of biology who's been involved in the intelligent design debate internationally for 3 decades. His grandfather, Byron C. Nelson, a theologian and author, was an influential mid-20th century dissenter from Darwinian evolution. After Paul received his BA in philosophy with a minor in evolutionary biology from the University of Pittsburgh, he entered the University of Chicago, where he received his PhD in the philosophy of biology and evolutionary theory. He is currently a senior fellow of the Discovery Institute and adjunct faculty in the Master of Arts program in Science and Religion at Biola University. As an early associate of Professor Philip Johnson of UC Berkeley, author of the best-selling critique Darwin on Trial, Paul was an organizer of the Mere Creation Conference in 1996, where the modern intelligent design research community first formed. His research interests include the relationship between developmental biology and our knowledge of the history of life, the theory of intelligent design, and the interaction of science and theology. Paul lectures frequently at colleges and universities throughout the United States and Europe, has spoken on American and Italian national public radio, and written for popular publications as varied as the Oslo Dagbladet. That's a newspaper, right, Paul?
It's their main daily paper in Oslo.
Yep, and the Christian Research Journal. Nelson's scholarly articles have appeared in journals such as Biology and Philosophy, Biocomplexity, Zygon, Rhetoric and Public Affairs, and Touchstone, and book chapters in such anthologies as Mere Creation, Signs of Intelligence, Intelligent Design, Creationism and Its Critics, Darwin, Design and Public Education, and the Next Generation Systematics, Paul is one of the authors of the biology textbook Explore Evolution and has appeared in several films on intelligent design for Illustra Media, and I've posted a couple of clips of those on our Facebook page. He is a member of the Society for Developmental Biology and the International Society for the History, Philosophy, and Social Studies of Biology. Paul is married to Suzanne Nelson, a pediatric gastroenterologist at Northwestern University, and they have 2 daughters. And the family resides in Glenview, Illinois, which is just down the road from where I am in Libertyville. So it's really a privilege to have you here, Paul. And like I said, I've been excited about this meeting since you agreed to come do the talk. The title of Paul's talk is The Promise of Design Triangulation for Biological Discovery. We'll do a Q&A after the talk. The talk's going to be about 45 minutes, more or less. We'll do a Q&A after that. I watched a couple of Paul's talks from other venues. I would tell you, buckle your seatbelts. It's really good. So welcome, Paul. Thank you again for being here, and I'll just turn the floor over to you.
Wonderful. Well, thank you all for joining. I feel like I live a lot of my life now on Zoom. So the software is getting more and more familiar to me. Can you see my title slide? You all good?
Yep.
Okay. So just an outline to let you know where we're going. I'm going to start with an everyday example of what I call functional triangulation. And this actually happened to me a few years ago when I was driving my daughters downstate for a badminton tournament. I could not get the left sliding door of my Honda Odyssey minivan to open. So there's a mystery there. Why wouldn't that happen? Then we'll segue to functional triangulation in biology. We'll look at the luckiest guess ever in the history of science. And of course, it wasn't a guess at all. It was a rational inference from Francis Crick. Then we'll shift to why functional triangulation is really design triangulation, and a notion I call the causal primacy of the organism. And then to conclude, parts, wholes, and the origin of life. And then Q&A, of course. Okay, so brief introduction. I met Scott Minnick when I was a graduate student. He is a microbiologist at the University of Idaho, a wonderful, insightful scientist. He does a lot of important work on Yersinia pestis, which is the bacterium that causes plague. He actually has worked for the US government as a specialist in that area. When we met, he told me something I've never forgotten. He said, Paul, this intelligent design debate really should not be that controversial. The fact is, the reality, he said, is that most molecular biologists are de facto design theorists already, whether they know it or not. And he said it's been that way for a very long time. Now, what struck me about that remark was, in my own life, before I had met Scott, I had seen this to be true. In the undergraduate courses I had in genetics and molecular biology, it was clear that the scientists in question were making design inferences, but not by that name. So he reinforced something from his own experience that I had seen in my education. But the puzzle is, how could that be possible when design was widely seen as intrinsically unscientific? Because as it's expressed in the idea of intelligent design, and it seems to refer to a transcendent cause, a mind that we cannot see, that's unobservable in principle. So in this talk, I want to explain what Scott meant by molecular biologists being de facto design theorists. And I want to use this idea of design triangulation, which I hope will be intuitively clear to you. I think many audiences have found it so, and I hope you do as well. Here's what we're not going to do. We are not going to critique evolution. Jerry Coyne, who's attacked me on his blog, I knew him at the University of Chicago. He's a professor of genetics there. Or Dan Dennett, or Dawkins. Actually, we will mention Dawkins very briefly at the end, or Jack Shostak, the origin of life researcher at Harvard. And that's not, I'm not going to critique them, largely because I think intelligent design ultimately needs to stand on its own two legs. And debunking somebody else's theory does not ipso facto support your own, okay? Now, I think all of these guys are interesting. I love reading their work, but I really don't want to focus on evolution tonight. And as I said, there'll be one brief mention of Dawkins at the very end. And if you want to know why, really, I don't like critiquing evolutionary theory, read C.S. Lewis's The Last Battle. In particular, chapter 13, there's this episode with quarreling dwarves. I won't say more about that, but it's very telling. It's a wonderful metaphor for what goes on in evolutionary theory with respect to the evidence for design. All right. This talk is an excerpt or a précis of a much larger file. Okay, so this talk is about 79 slides long. The larger file is over three times that long, and it has many more interesting details. I was hoping to have it available at the Discovery Institute webpage today. They're too busy right now, but Mike will be able to give it to you, or you can contact me. I actually have it loaded up on this laptop for the Q&A if you want. But I mentioned this because this is sort of an executive summary, if you will, of a much larger treatment of this idea of design triangulation. And I encourage you, if you're interested, contact me or contact Mike and get the big file, because this is more like a snack and that's a full meal. All right, why won't the sliding door open on my Honda Odyssey? Okay, so I actually don't own this van anymore. Now I drive a Honda Pilot. I love Hondas. They just run and run. All you have to do is change the oil. But when my daughters were in high school, this was the car that I drove. Okay, so you put me there in the driver's seat. Now, when they were in high school, they were both on the varsity badminton team at Glenbrook South High School, and I would take them around and their teammates to various tournaments. So imagine this scenario. I pull into a gas station and I ask the girls in the car, the young women, do you need to use the bathroom? Okay. And everybody says, no, we're fine, Mr. Nelson, no problem. So I pull into the gas station. We're on our way downstate. I pop open this, the fuel door. Okay, I've got a release up here where I'm sitting. I pop it open. And I want to get out to fuel the car. And one of the girls sitting back here says, oh, you know what? I do have to pee. Can you let me out? And I try to open this door and it won't open. Why not? All right, there's a clue. Actually, here's a clue. I only look at the owner's manual in my cars when I have a problem. Okay, otherwise it sits there in the glove box. I don't mess with it. So I can't get the door open, and I grab the owner's manual, and it says, make sure the rear sliding door is closed, and here's a clue why. All right? I'm very dim. You know, I may have a PhD in the philosophy of science, but in much of my life, I'm very dim. So I say to the Honda Corporation, why do I need to make sure? Well, two macroscopic objects with mass, like doors, cannot occupy the same location in space at the same time, right? It's just not going to happen. This door, when it's opening automatically, slides to the back. If the fuel door is open, there's going to be a collision right here, okay? The Honda engineers have anticipated the stupidity of Paul Nelson. They know that some drivers are clueless like me, right? So in the design phase of the vehicle, they say, we've got some clueless drivers who are going to try to open that left rear sliding door when the fuel door is open. So this is actually a screen capture from the owner's manual for the Odyssey, it's quite clear. If you release the fuel door, the driver's side sliding door will automatically lock. Okay, so I'm sitting there in the gas station. One of these badminton players says, I have to go, I have to go. So I push the button, intending to send a signal to open that door, and there's a logic circuit in the car somewhere. Is the fuel door open? Yes? You can't open it. Okay, now, I understand this because I went through it. My brother Peter, who is a researcher in kidney disease, is much smarter than me and much more clued in. He doesn't know anything about the Honda Odyssey, right? So he's looking at the car in my driveway, waiting for me to come out, and he studies this arrangement. He realizes, again, two macroscopic objects with mass cannot occupy the same space at the same time. And he says, Honda must have built in a mechanism to prevent the door from jamming. Okay, he triangulates. He makes what I'm calling a functional triangulation. This is the everyday version. Sliding door moves to the left rear over the fuel door. If that door were open, the sliding door would jam. Ergo, he triangulates to the existence of something he hasn't actually seen, but he has good reason to believe is there. Namely, the engineers of the Odyssey must have built in a locking mechanism to prevent the door from jamming. Now, this is not strictly speaking an existence proof, but it does confer a high probability that something like C is operating. Okay, you have done this in your own life. You've observed A, you've observed B for some complex system, and you say if A is the case and B is the case, then something like C probably exists. You just haven't given it a name, but you've made that inference. I'm calling it functional triangulation. Now, we can also use this as a metaphor for scientific inquiry. We know A, we know B, something like C is probably the case. But I want to apply this now to biology, all right? But we do this for complex systems all the time, where A and B are two facts about some black box. And we say, well, we're pretty sure about A, we're pretty sure about B. Something like C is probably true, even if we haven't yet observed it. Okay, now I've pointed out you've made this inference, you just haven't given it a name. Now we have warrant to go looking for C. We have good reason to think it's there. And this is a research motivating heuristic. This method actually would work really almost in any imaginable complex system where there are functional relationships that exist within that black box. So let's see how it works in biology. We'll do a thought experiment. And I've actually done this with students, and it's amazing how quickly they can make the triangulation work. Okay, so let me give you a couple of facts. And the black boxes in this case will be not just any complex system, but organisms. Okay, fact A, or number one, copper is a poison. There's a Bohr model of the copper atom. You do not want otherwise unemployed copper ions sloshing around your biology. They're highly reactive. They're toxic, right? A, copper is a poison. B, or two, copper as a metal is absolutely required by aerobically respiring organisms like you as an essential cofactor in several enzymes. Okay, this is well known. If you don't have a certain amount of copper in your diet every day, you will get very sick, because copper, here is the cytochrome C oxidase, which is a membrane-bound protein in your mitochondria. It's essential in the electron transfer process of the mitochondria. Here you can see there are copper atoms here, there are copper atoms here, and they're essential for the function of this protein, which, if I had another figure, you would see is embedded in the membrane of the power supply for you, your mitochondria that are producing ATP. Copper is an essential cofactor. You have to have it, okay? And there are many other enzymes where copper is involved. Now, You can do this. Free copper is a poison. We know that. You have to have it. It's absolutely required. These facts, because they both refer to copper, bear on each other. We can put our black box in here of human biology or mammalian biology, or really the biology of any aerobically respiring organism, and you can triangulate. Something must exist inside that black box if A and B are the case. Now, normally when I do this with the classroom where I'm in the same room as the audience, I get very literal. I make my fist into a copper ion, right? I say, here's my copper. It's poisonous. What do I have to be able to do? I say to the class, think very literally about this. And they say, well, you have to hold on to it somehow. Right. I've got to bind it, right? In a safe way. But the enzyme where it's a cofactor is over there in the cytoplasm. It's not here. Now what do I have to do? Well, they say, well, you got to move it, right? I've got to transport it. But when I get over to that enzyme, I'm still binding it. Now what do I have to do? And they say, well, you got to let it go in a controlled way. They triangulate to the existence of something they've never seen, but they know must be there. There's got to be a cellular system that binds, transports, and releases copper where the metal is needed. Now, imagine me in a conversation with a skeptic of intelligent design. We're in Starbucks, let's say, maybe outside Starbucks because it's COVID-19 era, right? So we got our masks on and we're talking. And he says, Paul, how can you be sure? This is how I can be sure, right? It's there. You can call this the no magic principle. Organisms make use of mechanisms to exist at all. Whether we see those mechanisms or not, they are there. You are not running by magic. The purging of magic from Western thought in the scientific revolution, not entirely because it's still around us all the time, magical thinking that people have, but the purging of magic as a serious pursuit of intellectuals trying to do natural philosophy was essential to the scientific revolution. The no magic principle is necessary to get to knowledge, all right? So whether the mechanisms are there, whether you've observed them or not, you can be sure that they're there. Now, what's interesting about this particular thought experiment is cell biologists knew many years before they actually observed the system that it had to be there. So in this paper in Science, actually it's a news report about a major discovery about the copper system. These authors say, look, it's a paradox. You have to have it, but it's highly toxic. So this contradiction was rationalized by assuming that copper, like other redox active metals, was sequestered safely in non-active forms and moved through cellular compartments. But they go on to say, look, we didn't know what was actually doing that job. We knew something had to be there, but until recently, the actual actors, the agents of that trafficking were unknown. What's interesting is by triangulating, they knew that system had to be there. That was warrant to go looking for it. And here it is. So here are the copper ions outside the cell membrane. They go through and they are shepherded through the membrane by a specialized protein, captured once they're inside, moved by a very complicated system to the site where they're going to be cofactors. And by the way, I can provide all the citations from this talk, the actual articles, if anyone wants them. Now, I love this language. This is a report in Science in 1997 describing the discovery of this system. And the authors use language that is highly suggestive of intelligent design. What they point out is they say, our results underscore the idea that cells make use of elaborate machinery for recruiting, trafficking, compartmentalizing, and ultimately inserting into the appropriate enzyme reactive cofactors such as copper ions. I love this. This is unlikely to proceed by spontaneous self-assembly. Preach it. It's an orchestrated event. So the very language here is descriptive of a design perspective, whether the authors are conscious of it or not. This works. This works. So when you get the big PDF, there are many other examples, both from the history of biology and today, where you can see this triangulation method in action. But step back for a moment and put on your philosopher cap and ask, why does it work? Why does this triangulation method work? What must be presupposed to get the inference started? Okay, I'll come back to that question, but it's important to think about it, because it wouldn't work for lots of systems, but it does work for organisms. All right. Now, this next example is the most telling, and it also carries the answer to the question, why does functional triangulation work, and why should we call it design triangulation? So, the luckiest guess ever in the history of science. Of course, it's not a guess at all. It's a rational inference using the method I've just described. Okay, forget any molecular biology you've ever learned and put yourself back in the mid-1950s, 1955 to be exact. At the time, Watson and Crick had solved the structure of this gorgeous molecule. But notice something interesting about DNA. It's chemically rather boring. You have a sugar phosphate backbone, like the legs of the ladder, right? Two of them coiled on themselves in the spiraling double helix with hydrogen bonds from the nucleobases as the rungs of the ladder. And it's just more and more and more of that. Repetition of sugar phosphate backbone, nucleobases with hydrogen bonding. Over here, by contrast, you have 8 of the 20 amino acids known at the time, and they're very different. Little glycine is much different in terms of its geometry from big phenylalanine, right? These are very different chemically. This is quite regular chemically. So Francis Crick looks at this and he says, I know that DNA carries genetic information. I know that proteins are built of amino acids, but how is this very regular geometry on the left, sugar phosphate backbone, hydrogen bonds, specifying or picking out these very different amino acids? I've only illustrated 8 here, but there were 20 at the time, 22 now. 2 are added post-translationally. What's mediating information transfer from one chemistry to the other? Okay, so characters in the story. Francis Crick will be the protagonist, one of my scientific heroes. I regret that I never was able to meet him because he taught me so much. And even though he was an atheist, he was a much better biologist than he was an atheist. Let me put it that way. Really, really, truly a hero of mine. And the antagonist, James Watson, who is honest to a fault about himself, brutally honest, okay? So these are going to be the 2 main characters in our tale. All right, in the early 50s, the physicist George Gamow, who was important in the Big Bang Theory and in development of atomic weapons, a brilliant man, fell in love with molecular biology. It didn't have that name at the time, but Gamow was particularly interested in the puzzle of coding. This puzzle of how information moved from DNA to amino acids in protein assembly was an open puzzle in the early to mid 1950s. And he started something called the RNA Tie Club, which was, in a sense, an invisible college. It only had about 24 members, and they all had ties with RNA on them. It was a very exclusive group. Francis Crick wrote a manuscript that was distributed to the RNA Tie Club that was never published. You can download it. The URL is there. And by the way, I'll give you the bibliography for this talk as well if you want to pursue these references. Crick wrote this manuscript, circulated it to the RNA Tie Club, but it was never published. And it's a shame because it's probably the most brilliant thing he ever did. All right. In that manuscript, he was trying to answer this question: how is genetic information passing from this chemistry to this chemistry? And he puts it this way. He says, what I find profoundly disturbing when I look at DNA or RNA is no one would think of them as a direct template for amino acids. Why not? Because they don't have the right geometries in 3 dimensions to isolate particular amino acids. He says, where are the knobbly hydrophobic surfaces to distinguish valine from leucine and isoleucine? Where are the charged groups to go with the acetic and basic amino acids? What DNA does show, and RNA will do the same, is a specific pattern of hydrogen bonds and very little else. All right, as I said a moment ago, Crick was a much better biologist than he was an atheist. And what he did, and I don't want you to miss just how audacious and brave this is. I've taught this episode for years, but every time I discuss it, my sense of admiration for Crick's genius and his courage never diminishes. Because he says, organisms are not working by magic. Something must exist to do that job. And the principle that he's using is right here. When you observe a complex system performing specialized functions, there's a rational logic and well-matched parts that enable those functions. No magic, look for the mechanism, it's there. So he triangulates. DNA carries information, that's our fact A, but is chemically nonspecific. Hydrogen bonds, little else. Fact B, amino acids have specific geometries which require a system to recognize them. From A and B into that black box of the cell, he says there must be some mediating or adapter molecules that enable information to move from DNA to amino acids. All right, we have the warrant to go looking for the system. And by the way, we've got 20 amino acids, so we need 20 of these specialized molecules. He triangulates. Each amino acid would combine chemically. And by the way, don't forget, this is 1955. There is no evidence for what he is describing. This is a leap in a sense, although it's not really a leap at all. It's a sound inference. This is an inference into space, into that black box, saying this has to be there, all right? Each amino acid would combine with a special enzyme that would bond at one side to nucleic acid and at the other end to amino acids. And we would need 20 of them and 20 enzymes to attach the amino acids. So Sidney Brenner, they shared an office in the 50s, in the late 50s. And Brenner had called this the adapter hypothesis, because you're adapting each amino acid to go onto the template. So let's take just valine. Remember, no evidence for any of this. He says we need that mediating molecule. At one end, it's going to bind to nucleic acid, right? It's going to pick up a signal over there on the left. At the other end, it's going to bind to the adapter, and the adapter will bind to a specific amino acid. To do that, you need a dedicated enzyme. This enzyme will have to know what this signal is. In some way, chemically, know what this signal is and say, oh, that's the signal for valine. That's my job, to attach valine here. And you're going to need 20 of these. All right. Now, Crick in the very same manuscript says, well, there's no evidence for this. But you know what? His courage is such that he just slaps that away with the back of his hand. He says, the usual argument presented against this scheme is that no such small molecules have been found, but this objection cannot stand. The inference from systems level functional necessity is very strong. This is the insight of a biologist who knows a system intimately, is not going to say it's running by magic. He knows the mechanisms are there, and he says we can triangulate to them. And here they are, right? Transfer RNA. Notice it has all the, and this was isolated and characterized many years after Crick predicted its existence. And it has the features he said must be there. Here is the anticodon, which is the nucleic acid binding surface, right? It's reading messenger RNA here. So what's interesting about this molecule is there's a greater than 70 angstrom distance from the anticodon to the CCA stem where the amino acid attaches. This is a true code. This means that there is no direct chemical interaction. So they knew it had to be a code, right? Because you're transferring from one kind of chemistry to another. The chemistry is not doing this. What's doing it is a semantic convention, namely, this signal here is going to mean that. And even more remarkably, amino-acyl tRNA synthetases. So here's the anticodon stem. The signal is here. And this very large protein is reaching down underneath to read the message. And up here is where the amino acid is going to be attaching. All right, none of this existed. There was no evidence for it at all. Crick knew it had to be there. Now, why is Watson the antagonist? The brutally honest antagonist of this story. All right, so this is the second part of the episode. And it takes us into why design triangulation is a better name for what's going on here than functional triangulation. All right. As you probably know, James Watson throughout his life has said things that are very impolitic about race relations and the abilities of women in science and so forth. He's probably somewhere on the spectrum. He's just brutally honest. And in this autobiography, published in 2002, he explains why he rejected Crick's idea. And he did reject it. So he's describing this period in the mid-50s when they're trying to figure out how information transfer is actually working. And he got this manuscript. He was a member of the RNA Tie Club. Crick sent him the manuscript. He had it, but he didn't buy it. Why not? Well, the interesting thing about his reason is it really had nothing to do with the evidence. The evidence at hand strongly supported Crick's hypothesis. And in fact, it turned out to be true, right? Transfer RNA is necessary for every living thing on this planet. Watson didn't buy it because it was impossible to fit with his ideas about the origin of life. The adapter mechanism seemed to me too complicated to have ever evolved at the origin of life. In other words, his biological intuition, unlike Crick's, was chained to an implicit time axis, like this. This is what is in the back of James Watson's mind. There is an arrow of time, and cells lie well along that arrow of time. There's a period in Earth history where there were no cells, then the parts accumulated somehow, RNA formed somehow, membranes formed somehow, then cells got up and running. This time axis has captured his biological imagination, and within the naturalistic picture of life, complex biological systems cannot arise all at once. They're just historical entities, and history is just one damn thing after another, right? So when he thinks about the origin of cells and the origin of the DNA, RNA, protein system of information transfer, it's too much at once. Biological complexity can only accrete over time slowly. So he rejects the adapter hypothesis, and in a sense, he's a better atheist than he is a biologist, because this had to be there. Once the facts about DNA were known, once protein assembly was understood in terms of amino acids with their very different geometries, something doing the job of transfer RNA had to be there, and Crick knew that. All right. Now, step back again, put on your philosopher hat. Where does that time axis come from? Where does that premise that the complexity can only accrete come from? They're not there in the biology itself. The premise, simplest at the start and only then more complicated, and one must begin with the parts, those come from naturalism, from a naturalistic metaphysics of explanation, from philosophy in the sense that the Apostle Paul used it as a pejorative. The evidence itself from organisms does not sustain those assumptions. They come from a naturalistic philosophy. Actually, to learn things in biology, this method works. And when you get the big file, I've got a bunch of gorgeous examples in there, both from contemporary biology and going back into the history of biology. This method works, but it does make a demand on you as an investigator. You have to presuppose the prior existence of the system as a whole, because that's the only way you can get the functional necessity relations hidden in that black box that warrant triangulation to the parts you haven't seen. You've got to have the black box put together and working to make these triangulation inferences. So one last thought experiment to illuminate this point. And I can't believe it. I'm going to finish on time. Praise God. All right. Parts, holes in the origin of life. Okay. Here's a black box right out of Google Images. Okay. A friend gives this to you and he says, just put it on your nightstand and watch what happens. Okay. So I'm going to give you this black box. Not very big. Put it on your nightstand. Watch what happens. All right. The next morning, 8 a.m., you observe the following. On even numbered days, like yesterday, October 12, it plays Bach. Jesu, Joy of Man's Desiring, the Goldberg Variations, right? Some Brandenburg Concerto. It's playing Bach only on the even numbered days. On odd numbered days, like today, plays Beatles. So this morning it played Hey Jude. The box infallibly keeps track of the even versus odd days, depending on the calendar, and it always plays Bach or Beatles, depending on what the value of the date is. Okay? You watch this for a week. You happen to like Bach, you happen to like the Beatles, so it doesn't bother you that much. And you start thinking, what's got to exist for this to happen? So what are you starting with? You're starting with the system as a whole and these kind of curious functions that it performs. That's your starting point, all right? From the system as a whole, you can infer to the parts you haven't seen. Something is keeping track of the time and the calendar. Something is storing the music of J.S. Bach and the Beatles. Something is scheduling the music depending on what's the case here at 1, all right? And there's the basic housekeeping stuff. You got to have a power supply, amplifier, speaker, and so forth. Now, pay attention to the logic here. In other words, what's primary, right, in this causal inference that we're making? What's inferentially basic? Well, you know what? It's not the parts. And you can prove this to yourself. If you don't know the target state first and the functions it performs, you'll never get to the parts, or you'll never get those from the parts, right? There's a logical asymmetry here. Now, in the big file, I go into this in a lot more detail with other examples, and I'm sorry, I'm going to have to kind of scoot over some of the philosophical terms here, but the inference from the black box to its parts is possible, but you can't go in the other direction because the space of possibilities is too large. So if I gave you this bunch of circuits, transistors, resistors, speakers, and so forth, and you didn't know the target state, you didn't know that black box I just described, you would never be able to get back to it, because the range of possibilities represented by these parts doesn't entail those unique functions. You can go lots of directions from these parts, all right? The range of possibilities is too great to go from the bottom level up to the top, but the other direction is possible. You can go from the higher level system and triangulate to the parts that must be there, even if you haven't seen them yet. That logical asymmetry turns out to be very important for biology. Now, this is just a thought experiment, but you can do this with any number of examples. In fact, the last time I gave this talk, somebody emailed me and he said, the best example for this, Paul, are those big Lego sets. What, you know, the ones that cost hundreds of dollars. You can go on the web and get a list of the parts, hundreds of them, thousands in some cases. You can't go from the individual Legos if you don't know already what the target state is. You can't go back to what you're supposed to build. But you can go the other direction. So in the big file, there's a Lego example. I grew up playing with Legos, although they weren't the souped up Legos we have today. Anyway, you can do this. You can't go the other direction because the parts don't uniquely specify the higher level system. For biology, this ends up being very important. All right. Let's look now at a biological example. I'm calling this the causal primacy of the organism. Organisms are loaded with clues. In effect, nature calls out to us. She gives us what we need to make these inferences because organisms, even single-celled bacteria, have this complex web of functional relationships that enables us to make these inferences. This is what Scott Minnick meant when he said most molecular biologists are de facto design theorists because they're working their way around this web of relationships and discovering connections that have to be there. And in the big file, I discuss multiple lines of evidence that have accumulated over the last 4 decades that point to this, the causal irreducibility of the cell, okay? Now this paper, and I'll send this to Mike, and he can make it available to you. This paper is truly beautiful. All right, when I was a kid in my late teens, I wanted to be a painter. And I went to art school for 2 years and I was so miserably unhappy. I finally said to God, all right, you don't want me to be a painter. What do you want me to do, right? And I discovered the beauty of science. I never knew that before. I never knew that science could be as beautiful as music or poetry or art, but it really can be. And this paper is truly beautiful. It's by 2 protein biochemists describing what they call the Leventhal paradox of the interactome. This is the shadow of a science yet to be born. In the same way that the work of Carnot on steam engines in the 19th century foreshadowed the birth of thermodynamics later on. It's a really elegant paper. And what they do is they say, or they ask the following question, take the parts of a cell. Okay, this is the interactome for Mycoplasma genitalium, a very stripped down bacterium that in its normal environment is an obligate parasite of the epithelia of the human airway and our genital tract. Notice the name here. Now these are proteins. Each of these lines here with a number is a protein. So these are the proteins that are involved in the ribosome, the translation system. It's a big collection because that's a big machine. But you've got proteins involved in cell division, proteins involved in energy metabolism, and so forth. Okay, think about this like a parts list. And each of these parts is an individual entity. And they interact. All right? My picture back here. They're interacting in this web of causal relationships. That's what they mean by the interactome. And what they point out is, as the number of parts goes up arithmetically, their possible interactions go up exponentially. Okay? What that means is, imagine you have a single bacterial cell in a sterile buffer. All right? It's alive. It's floating there in a sterile buffer, and you hit it with sound. One of the ways you can sterilize things is using a method called sonication, where you hit it with very high frequency sound that disrupts the cell membrane. At that moment, when the cell membrane is disrupted, every bit of hardware is there. Yet the cell will never come back to life, even though it's kind of the glorious dream of every origin of life researcher to have a complete chromosome, ribosomes, lipids, all the hardware that you need for a cell. You disrupt that network of relationships, and what happens is all the parts begin to interact in ways that have nothing to do with the living state, and it scales exponentially. So here you see the number of parts going up arithmetically, and on the right-hand side, you see the possible interactions. Now watch how that number gets bigger. Bill Dembski did these calculations for me. So here we're at about 50 parts, 49 parts. The possible interactions are sailing out of sight. And when we get to 100, it's just an absurdly large number. Bill stopped here. I asked him to do these calculations for me. He stopped at 100 proteins, and he said, Paul, this number is just ridiculous. All right. The possible interactions among those parts that do not yield the living state is astronomically larger than the very tiny neighborhood of interactions where the cell is actually alive. So he didn't even bother to go past this point of 100, even though real cells have at least 300 proteins. He said there's no point in going any further. Now, you know who knows this? Richard Dawkins knows this. It's not generally appreciated just how much of an intelligent design thinker Richard Dawkins is. But in The Blind Watchmaker, he said, however many ways there may be of being alive, it is certain there are vastly more ways of being dead, or rather not alive. Because he realizes the matter, the mass, the bits and pieces that go into a living thing have to be in that very special neighborhood of alive. And surrounding that neighborhood is an enormously, unimaginably larger neighborhood of not being alive. So here's our space within our space of possibilities. Inside is a live bacterial cell. It's alive in there. This is a tiny, tiny neighborhood. And the Leventhal paradox of the interactome, I am not exaggerating. It is the shadow of a science of biological design yet to be born, describes why this is a paradox, because you can have all the parts, but unless they bear the right functional relations to each other, you're going to go out and wander irreversibly in the space of not being alive. So take our little box here, and let's put it in a space that would approximate what it means to be dead. All right. There's Andromeda. Very nice picture of Andromeda, our neighboring galaxy to us, billions of stars. Here's our box, and it's lost somewhere in that space. And you will never find it unless you know where it is already. You will never construct it bottom up. The authors of that paper, Tampa and Rose, are not design theorists. I don't want to misrepresent them, but they have described a biological reality that really only makes sense from the perspective of top-down causation, that cells never arose bottom up. When you get the paper, and I'll send it to Mike so he can distribute it to you guys, you'll see what I mean. Why does this work? Because you can start with holes. And biologists who do that are actually borrowing a premise from design, whether they're conscious of it or not. This is the metaphysic that enables that to work. All right, I'm done. I finished on time, which is a miracle. Miracles happen. So please, if you get the bigger file and you see something that doesn't make sense to you, email me and I'll try to get back to you. But it really is, this was a snack. And the bigger file, 3 times the length, is a full meal with a lot more examples and discussion of the history and philosophy of these questions. And I'm sorry, I just didn't have it, was not able to get it posted at Discovery Institute in time for tonight's presentation. So we have time for some Q&A. If anyone...
Yeah, and no worries on that, Paul. If you send that to me or send me the link, as well as the article, I'll be happy to share this with
I will definitely send it to you.
Yes,
I actually have a note right here on my laptop.
Okay, awesome. So yeah, we'll open this up to Q&A. Paul has a hard stop at 8:20, but I think we should be okay. All of you are on mute at the moment. So when you have a question, if you will remember to unmute your mic and ask away, that'd be great. I know I've seen a few comments on the Zoom group chat, and so perhaps questions come out of that. While people are thinking of questions, Paul, I'll start with one. So you mentioned Richard Dawkins, and here's a smart guy that has got to know the empirical data for the things that that you've discussed tonight, and yet rejects the idea of intelligent design. Is that a philosophical commitment that he has? What is it, I would say, tongue in cheek, is he deluded?
Well, think about evolution in a comparative sense. So compare the theory of evolution to other scientific theories, like the wave-particle duality of light, or plate tectonics, or the germ theory of disease, and so forth. Uniquely, of all scientific theories, evolution is the theory about us. It's the theory about where we came from. And in that respect, it has never been just another scientific theory. It functionally takes the role of a worldview.
Right.
So some of the scientists I admire the most in the current community, such as Eugene Koonin at the NCBI, National Center for Biotechnology Information, brilliant Russian theoretical biologist who came here as the Soviet Union was collapsing and now runs a big lab there. And I read just about everything he publishes, okay, because it's so full of ideas and good information. When confronted with some of the puzzles I described in this talk, in particular, the origin of the information transfer system, he said unmistakably, this will never happen by normal evolutionary processes. All right, so he walks right up to the edge of a design inference. But what he decides to do is to appeal to chance. And I'll make a note, Mike, I'm going to send you this paper because it's so illuminating. What he does is he says, let's make the universe infinite in temporally infinite and spatially infinite. When you do that, you can swamp any small probability. So you can win any lottery if you buy all the tickets available, right? Because you swamp the small probability that you're not going to win. You get all the chances in your own pocket, and you're bound to win because you bought all the tickets that were possible, right? So even though the probability is very small that any one person will win a lottery, that's how they work. If you're crazy enough to spend all your money, and billions of dollars more and buy all the tickets, you're going to win. So what he does is he buys all the tickets using infinities, and he says, even though we can't imagine how this could have happened by a natural process, it could have happened by chance, by this move. All right, at that moment, what Eugene Koonin has done is he's left science, not for evidential reasons, for philosophical reasons, because the alternative is design. And I think we need to be very candid about the fact that science interfingers with philosophy and theology when we come to these questions of origins. So I admire Dawkins tremendously. When I taught writing at the University of Chicago, I was a graduate student, and I had the job of teaching in the professional writing program there. I would use the chapter on bat sonar from The Blind Watchmaker because it was so beautifully written. It could, just this beautiful exposition of the amazing design of bat sonar. So Dawkins appreciates the beauty and design of living things, but he does not want to attribute that to a transcendent mind, which is, When I began working with Bill Dembski and Steve Meyer on intelligent design over 2 decades ago, we realized that Mother Chance is always there with her arms wide open to hold you in an irrational embrace. And you can't argue against chance. Ultimately, it's a choice of the individual. And Dawkins chooses that.
Right. So let me just stop and I'll be quiet, and anyone that is in the meeting.
Yeah, Mike, this is Steve Jurich.
Yes, Dave.
Good. I really enjoyed the talk. Exceptionally complicated topic. So I had a couple of questions that I'm not quite sure how this is going to play out, but the things I was curious about based on your talk. One was, first one was on triangulation, right? So you looked into protein synthesis, right? Then what you didn't touch on there was chirality of the original amino acids. So how would that fit into a triangulation context? You've got a specific chirality of amino acids, and there's not a lot of data to suggest why one chirality is preferred over another, or even how it originates. How do you triangulate back to single amino acids? So that's question number one.
Can I take
question number two? Entirely different subject. When you're looking at, and I'm a rational design person, but let's look at tumorigenesis. So I'll give you an example, say chronic lymphocytic leukemia. That can arise, most recent theories, from during the B cell differentiation process, right? So you just develop deletions in particular genes, or even hematopoietic stem cells, where you actually develop deletions or dysfunctions within the cells. That is not a rational process. I mean, that seems to be a process that's out of whack. So I would just sort of say on that, well, what's your thought about then the existence of processes which are, in fact, non-life sustaining and actually lethal processes? Two tough questions, I think, but I'd appreciate your thoughts.
Let me take the question about chirality first. When I was a student, I read Crick's beautiful little short book, Life Itself, published in 1981. And in that book, Crick is deeply agnostic about any hope for a natural origin of life on planet Earth. And he seriously proposes, along with Leslie Orgel, the notion of directed panspermia, that the reason that life appears more or less fully formed on Earth when it's first manifest in the fossil record is because life actually didn't start here. It was sent here by an alien intelligence. And Crick never disavowed that hypothesis. When you read that book, it's clear that Crick can see that the chemistry that's exploited by cells does not explain their origin, because the chemistry is indifferent to the living state. So when amino acids are produced by chemical processes, they're produced in hundreds, just in abundance, hundreds of different amino acids. Only 20, now 22, are found in living things. So somehow you have to get rid of all these other amino acids that will not give you proteins, as far as we know. And as you point out in your question, they have to be of the right-handedness, okay? Crick was a very good crystallographer. In fact, that's what he did his PhD in. And he realized there was no natural mechanism to discriminate these different chemical forms. So he resorts to a kind of atheistic intelligent design, directed panspermia. Life comes here from another planet, sent by an intelligence. And that's a case where I think his biological intuition, he should have been taken it more seriously. But again, the philosophical barrier there, I'm not going to infer design, led him astray. In terms of your second question, cancer is a real puzzle to me, because what you have is a system that's gone awry. And in a sense, it works, right? So in certain cancers, you have the formation of novel blood vessels that, strictly speaking, were not coded for in the normal biology of the organism, yet there they are, right? There's a vasculature feeding that tumor, yet they're going to take down their host. It's in the nature of cancer that these cells have gone rogue, and they are no longer part of the kind of republic of cells where everyone agrees we're all here for each other, right? The cancer has gone off on its own. It's going to monopolize resources. It's going to metastasize if it's a metastatic form of cancer, and eventually will kill the system that's sustaining it. So it's funny you should mention cancer, because for me, it's a giant question mark. And actually tomorrow and Thursday, I'll be in an online seminar on cancer and evolution to try to get some insights into how to understand cancer from the perspective of intelligent design.
Thank you.
So that, I'm sorry, I can't give you a better answer than that, but cancer is a mystery for me.
Yeah.
And as a Christian, I believe, of course, in the fall and the fact that we live in a world that is not what it was meant to be. But I'm being honest, I don't understand cancer from an intelligent design perspective.
Yeah, you and me both. Thank you.
Yeah, thank you, Steve. Others? Annie, you had some good comments on the chat. Anything to add?
Sure. On that cancer dysteleological, it's a dysteleological argument, right? So Paul, would you say, because to me, it doesn't, well, it brings up the problem of evil. It doesn't negate design, because only by recognizing a functional design in the first place can I then secondarily see that there's a broken design at work, right?
You're right. And that's where I would go as a kind of sort of first, the metaphor is I'm crossing a raging stream and I have to find the first rock where I can place my foot. That's the first safe rock that I can see, which is that it is a dysteleological system. It doesn't negate design, but it does raise questions about how can systems go awry and yet, in a sense, be functional, right? Because cancer cells are making a go of it. I'm not sure it's the right example. I know that there are cell lines, there are cancerous cell lines that have been maintained for many, many years, right, in culture. So I guess what I'm looking for is a well-articulated, intelligent design explanation of the formation of cancer, why it happens, when it happens, and that doesn't yet exist. Part of the reason is we don't yet have a fully articulated theory of biological design. Discovery Institute hates it when I say that, but I'm just being honest. And I think once we have a fully articulated theory, we'll be able to perhaps even predict why cancer occurs and where we should expect to see it. And there are clues, but the existence of bad design, and Annie, I appreciate the comment, does not negate the fact that there is real design, but definitely something has gone wrong.
Yep. Thanks, Annie. Anyone else?
So maybe a follow-up to that, because I've heard some in terms of why do things go wrong, well, here we go back to chance variation, right? But and I don't totally understand this very well. So really asking, because you were talking about, I think, Dawkins falling back on chance, but really isn't chance just no explanation at all? Isn't it like chance of the gaps? Like, isn't there always some actual kind of causality at work, and that chance is just this sort of smoke screen for we don't know, which is why I don't find, I'm with you that chance changes as an explanation for cancer doesn't quite seem to work for me.
Yeah. I do think that often chance is a cover for ignorance, and there is some causality going on. Again, from a deeper Christian perspective, this is Proverbs 16:33. The die is cast into the lap, but I'm sorry, I'm a little tired. I'm trying to remember the exact phrasing. I don't believe in chance as a real thing, because from the God's eye perspective, there is causality involved. And chance, I think, is always conditional on our lack of knowledge. Someone rolling dice in a casino there's a complex nonlinear process there. They're throwing the dice down the table. And if they knew exactly what was going to happen, there wouldn't be any interest in the game, right? So we appeal to these causal processes where we can't follow all the interactions, and then we assign a name to that of chance, even though the causal interactions are actually there. And if we had the God's eye perspective, we could understand them. For me, chance in an evolutionary context is a leap into irrationality. Because what you're saying is the evidence, like Kunin's paper, that's really so striking. He's very clear. He says the evidence here shows that this system could never arise from its parts. It would certainly never arise in a Darwinian way, because there's no function to select until all the parts are present. That, to me, is nature calling out to Eugene Koonin, saying, recognize the design that's here. But there's a philosophical cost to be paid, right? There's ultimately a spiritual cost to be paid. So Koonin opts for the embrace of infinity, basically. He says, make the universe big enough and it can happen just by chance, right? Biology class would be over in the time it takes you to write luck on the blackboard, because there's no knowledge to be conveyed. You just say, well, damn it, it happened, right? Here we are. DNA, RNA, protein system, here we are. Isn't it lucky? That, to me, is the end of, like the multiverse hypothesis, that is the end of science, because there's actually nothing to teach at that point. Certainly nothing to understand.
I'm mystified by the fact when you have two identical twins growing up in an identical environment, one gets cancer and the other doesn't. Is that God at work, Cain versus Abel?
Oh my goodness. You guys, these questions are way too hard for 8:15 at night. There comes a point for me when I have to have the attitude of a trusting child for some of these puzzles. And why something goes wrong with a pair of identical twins when apparently they have largely the same genetic constitution. There's a lot of noise in biological systems, right? And if you look closely enough at identical twins, monozygotic twins, you can find thousands of differences, but they're very minor, right?
Identical environment.
Well, maybe one twin liked rock climbing and they got way too much UV, and their DNA repair enzymes couldn't keep up with the work involved, and they developed melanoma, right? I mean, there are any number of circumstances where you can see why one would end up with a cancerous growth and the other not. But I don't know, you guys ask really hard questions.
No, that's good. Thanks, Thor. Okay, Paul, I want to be respectful of your 8:20 hard stop, and we're like 4 minutes from there. So maybe we'll wrap things up here. Thank you so much for being here again. Your talk was fantastic. I'll be looking for the
big PDF.
The big PDF, exactly. And I'll be
I am going to send some articles too. I've got some notes here. And please, if you have follow-up questions or things that I left unanswered, and I realize I'm a little bit on low cell this evening, I'm always just an email away. And Mike has my email. So I really enjoyed it. And I look forward to further interactions with all of you.
Paul, you are always welcome back here. In fact, I would tell you, I would love to have you back for another talk. I've listened to a number of them, and we just got an appetizer tonight in terms of what Paul has to offer. Let me close by saying that our speaker next month is Sarah Salviander. And I just think it'll be a fascinating talk. She is a cosmologist. Lee and Rebecca, she's in Texas somewhere. But she's a cosmologist who was an atheist, and her study of cosmology brought her into belief in God. And she's got an interesting story. She's not going to, we had Cy Gart tell his story in terms of how really the science brought him to faith. Sarah is going to talk about Genesis and how that is reconciled with science, and she's done some really interesting work on it, and I'm looking forward to her talk as well. Hopefully, the speakers that we have, at least it was the intention that we'll get different topics each month, and hopefully each topic will contribute something in terms of the foundation for your faith, and it's oftentimes it's a cumulative case, and certainly Paul did a wonderful job of that portion of the foundation. But we'll have people from different disciplines, different walks of life. December's former CEO of Baxter International, Harry Kramer, who is a Christian and is going to talk about his life as a Christian leader, and talk a little bit about a book he just wrote as well. So anyway, thanks again, Paul.
Thank you. I enjoyed it.
8:20, so
Thank you.
I'm gonna call this an end, and thank you everyone for attending. Have a great rest of the evening.